All right, welcome to another deep dive, everybody. Today we're going to be talking all about something you definitely need, but probably don't think too much about: vitamin B2,
Yeah.
otherwise known as riboflavin. Um, we got some pretty interesting technical information about how this stuff is actually made and used, and we're going to break it down for you.
Yeah. It should be fun.
Yeah. So, why should anyone even care about how a vitamin is made?
Yeah, that's a good question.
Right. Why?
Yeah.
Well, as turns out, the industrial production of vitamin B2 is actually like a window into some super interesting stuff. And we're talking about like biotechnology and the inner workings of our food supply.
Right.
We're going to be talking about the tiny organisms that make it all happen
Right.
and some of the surprising details about how they do it. We're really going to get into like the technical nitty-gritty of how vitamin B2 is actually produced.
So it's true. We definitely take these essential nutrients for granted. But when you actually look at how something like vitamin B2 goes from like microscopic life to a pill or fortified food, it's pretty impressive.
Yeah, it's really wild.
Yeah. And a testament to some clever science.
Yeah, for sure.
We'll be focusing on that journey of industrial production based on the details you sent over.
Okay, so the real heavy lifting here is done by microbes.
Yeah.
Tiny little things. We're talking about bacteria, specifically Bacillus subtilis. That's like the main character now for making riboflavin.
But there used to be fungi involved too, right?
Exactly. So historically, fungi like Ashbia gossypi and Erymathecium ashbi were really important.
Okay.
And E. ashbi is actually still used in some cases.
Oh, wow.
A. gossypi could actually produce a ton of riboflavin,
Nice.
which is great, obviously. But it created this super thick liquid that made it really hard to work with when you scale up production.
Oh, that makes sense.
Yeah. So what's interesting is how Bacillus subtilis kind of like rose to the top,
Yeah.
became the preferred organism.
Yeah.
It's super easy to manipulate genetically.
Okay. Meaning that we can actually tweak it to produce even more vitamin B2.
That's cool.
Plus, it grows quickly, and we've got well-established methods for fermenting it. So it wasn't just about the amount produced, but also like how practical the whole process is.
Makes sense. Efficiency.
Exactly.
So you've got these little tiny Bacillus subtilis. They're going into big tanks for something called submerged fermentation.
Mm.
It sounds kind of like they're swimming in a nutrient broth.
Mm-hmm.
So what exactly are they eating in there?
Yeah, so think of it kind of like a carefully balanced diet for these microbes.
Okay.
They need carbon for energy, stuff like glucose, or even things like molasses and vegetable oils.
Oh, wow. So they can break down those complex sugars?
They can, yep. And then they need nitrogen to build their cells, which can come from things like yeast extract or corn steep liquor.
Hmm.
And just like us, they need minerals, various minerals like phosphate and magnesium. But here's a really interesting part. The amount of iron is super critical.
Iron. I mean, we need iron.
Yeah.
How can it be so important, but also like something you have to control so carefully when you're making vitamins?
That's the really clever part.
Okay.
So for Bacillus subtilis, if you actually limit the iron in their environment, it actually pushes them to make more riboflavin.
Really?
It's almost as if they have all these building blocks ready, and without iron, they kind of get stuck.
Interesting.
And they just start pumping out the B2 instead.
That's wild.
Scientists can even add in stuff called iron chelators,
Okay.
which basically just grab onto the iron and keep those levels nice and low.
Okay.
There's another technique, too. You can limit something called purine nucleotides. That can have a similar effect, but that one's not as common.
Okay.
But overall, this really highlights a key principle in industrial biotechnology. Sometimes carefully controlling the environment can lead to huge increases in production.
That's so cool. It's like you're steering the bacteria, towards making more riboflavin just by controlling what they have access to.
Mm.
And this whole process happens under very specific conditions, right? Like temperature and pH.
Absolutely. It's a very delicate recipe. So for B. subtilis, the temperature's usually around 30 to 35 degrees Celsius.
Yeah.
And the pH needs to be slightly acidic to neutral,
Okay.
around 6.0 to 7.0.
Okay.
Oxygen is also essential.
Right, because they need to breathe.
Exactly. It's an aerobic process. So
Mm.
the tanks are constantly being agitated to make sure there's enough dissolved oxygen in there.
Right.
Often they use something called a fed batch system,
Okay.
where they're basically adding nutrients gradually over time to keep those microbes happy, growing, and producing efficiently.
So it's like a constant drip.
Yeah, exactly.
Okay, so now the bacteria have done all their work and you've got this broth
Yeah,
containing riboflavin
Yeah.
and a lot of other stuff.
right.
So how do you actually get the pure vitamin out?
So this actually depends on which microbe we're talking about. So Bacillus subtilis releases the riboflavin outside its cells, but while E. ashby actually keeps it inside.
Oh, so that's a big difference.
It is. So for B. subtilis, the first step is just to remove all that cell mass.
Okay.
And you can do that by spinning it down in a centrifuge or filtering it out. Then we can use methods like adsorption,
Okay.
where the riboflavin sticks to a material like activated carbon and can be washed off in a purified form. Or you can use something called ion exchange.
Okay.
For E. ashby, you actually have to break open the cells first.
Oh, wow.
It's a process called cell disruption.
Interesting.
And then you extract the riboflavin using solvents or water-based solutions.
Okay, so different microbes, different extraction strategies.
Exactly.
And after all that, I guess there's still more to do to get it really, really pure.
Exactly. We use further purification techniques, often more chromatography.
Okay.
Think of it as like a filter that separates out the different components. And because riboflavin doesn't dissolve very well in water, crystallization is actually a key final step.
Okay.
So by carefully controlling the conditions, we can get these pure crystals of vitamin B2,
Wow.
which are then separated and dried.
So it's a whole process. It's a journey, all this intricate work. Where does all this industrially produced vitamin B2 actually end up?
So most of it goes into making nutritional supplements.
Okay.
And to fortify foods.
Right.
So you'll find it added to tons of common foods like cereals and flour, milk, baby formulas.
Yeah.
All that helps to prevent deficiencies.
Right.
It's also used in pharmaceuticals as an additive in animal feed.
Oh, wow.
And it even has a limited use as a natural yellow food coloring.
Oh, that's interesting.
Beyond that, its fluorescent properties actually make it really valuable in research and diagnostic tools.
Okay.
And then it also plays like a really key role as part of these vital coenzymes in our bodies, like FAD and FMN.
It's amazing how much work goes into making something we might just swallow without even thinking about it. I mean, this deep dive has been really cool.
Yeah, it's pretty amazing.
It shows the power of using these tiny little biological factories to produce something so essential to our health.
Absolutely. I think that's the key takeaway here.
Yeah.
It demonstrates the huge role that industrial biotechnology plays in ensuring that we all have access to these essential nutrients.
You know, thinking about all these precise steps involved in creating just one single vitamin, it makes you kind of wonder about all the other hidden complexities behind other components of our diet, right?
Oh, definitely.
All the stuff that makes up our health. Something to think about.
Yeah.
All right. Well, thanks for going on this journey with me.
Yeah, this was fun.
Really appreciate it. And we'll see everybody next time.